Encore une fois, avant tout, c'est nos amis de Paris que nous voulons encore une fois remercier de venir ici. Nous avons une session fantastique, and I'll switch to English as usual, but we have three ladies, fantastic scientists, and I always believed that there is no need for anything special to give to women scientists. I remember Ruth Arnon 20 years or 30 years ago in a big meeting in Berlin. She fought against it because they don't need anything extra. They need just a little bit less discrimination. And those who think that today discrimination is finished are wrong. There is still a lot of discrimination. But in the meantime, let's enjoy three fantastic lectures by three fantastic ladies. And I start with Ada Yonat, who is still busy. But I must say, just a few days ago, I think in Nature, I found an interview with her colleague in Nobel Prize on ribosomes, who is now he's Indian and he's president of the Royal Society. And he said at the very beginning that everything was started by Ada Yonat. So Ada Yonat, now it's your turn.
Good afternoon. I hope that all of you recovered from that. As you can see, I slightly changed my title because I found out that the lecturer before me, Claude, talked to you about quantum mechanics, quantum physics, quantum. And I also remembered that this year we are celebrating 75 years for the book that Schrödinger wrote about life. So I want to give or contribute this lecture to the memory of Schrödinger. And just to remind you, he had a cat, and the story about the cat is part of quantum mechanics. So Schrödinger wrote a book. Maybe you don't really remember it, but he did. What is life? And actually he talked about the chicken and the egg problem. What was first? And I'll come back to it. But before I go on to this very important life questions, I want to show you several pictures. And I wonder, maybe the students or the professors know what's common to all of these. These are very, very important composers, philosophers, writers. What else is common? All men, yeah, correct. I'm sorry, I should have put a woman, but I'm not, I did not. And all Europeans, when I go to, and I show it in Asia, they tell me all European, all men. But all have nose, but something else that is specific. Sickness. You see that on their face that they are sick? So you're right. All of them are geniuses in their field. All of them died young. All of them died before the middle of last century, the middle of 1950. The last one was Orwell. And all of them, which you cannot see here, everything else you could see here, but you cannot see, is that they died from infectious diseases. And in my lecture, I will talk about the contribution of fighting against infectious diseases through ribosomes. So because of the fundamental role played by ribosomes, many antibiotics target them. Almost half of the clinically useful antibiotics target the process of protein biosynthesis. So you see here I am lucky twice. quantum studies were mentioned, and also proteins. Actually, all what we heard earlier is how they die, how they are taken away from the cell. I will tell you how they are made. All these proteins that Avraham Hershko already picked out. And the natural antibiotics are weapons that one bacteria, or one type of bacteria, is using in order to take care, to fight, to interfere with the life of other bacteria when they have, especially when they have problems, when they have competition about possibilities of life, food or area. So, as I said earlier, most of the antibiotics, or half of the antibiotics, are interfering with protein biosynthesis. And just a few words about proteins, more than what you heard before. First of all, proteins work only when they have correct fold. You can see here I'm using a very complicated machine, sophisticated machine, paper clips, that if their fold or their structure is being changed, they cannot do their very important job. And this is just difference in structure. It's the same, exactly the same paper clips. I didn't boil them, I didn't cut them, I didn't do anything to them, only changed their structure. And that's very important because the structure of proteins is determined by the sequence of their amino acids. These are the components of proteins, as you heard in the morning. And this sequence is dictated by the genetic code. And the genetic code are genes. You know what are genetic code? What is genetic code? You know what is DNA, young people? You sure? Shall I tell you what younger people say about DNA when they describe it to me? Really younger, 7 years old. They say DNA is the tool that the police is using to identify criminals. So that's also a way to describe DNA. But we are talking now about DNA in broader terms. And these are genes that are made of the 4 letters of DNA. Each combination of 3 of them is coding for an amino acid about which you heard in the morning, the components of proteins. So the structure of the proteins is determined by the sequence of the amino acids, and this is determined by the gene that codes for it. So the genes are located in the DNA. The DNA has many morphings, but it has also the genes. Here are the letters. They are in pairs, base pairs. There are only 4 letters. All biology, 4 letters. I'm still excited about it. And I can't believe it. But here the information is covered by the structure, the complicated structure. So there is a step called transcription where the information is encoded in a similar molecule that can stay as a single molecule. It also has only 4 letters. 3 of them are exactly like in DNA, one a little bit different. And this can be translated into proteins in every living cell. You heard about it in the morning, by ribosomes, by a machinery that exists in every cell, from bacteria, cockroaches, fish, flowers, human, ribosomes. So the main function of the ribosome is decoding and peptide bond formation. Peptide bond is the bond between amino acids that make the proteins. These are universal. They are performed in a very similar way, maybe even exactly the same way, we didn't prove it yet, in all cells, regardless of their source. And there is a huge number of ribosomes that function in each living cell. Some mammalian cells contain millions of ribosomes, like in the liver, 4 to 5 millions of ribosomes. Even bacteria cells, when they grow, they may contain up to 100,000 ribosomes. In vivo, it means in the cells, they act continuously, and they form up to 40 bonds in a second, and hardly make mistakes. Mistake rate is 1 to 500,000, or actually closer to 1 to a million. Hardly any mistakes in the translation. Now I just want to say a few words about 40 peptide bonds in a second. Here is something personal. I was the fastest student in school. In the lab, I was the first one to finish the labs when I was first and second class in university, because I was very poor and I had million jobs, so I had to work. And therefore, I did my lab work very fast. I needed 6 hours for one bond, the fastest in the class. And the ribosomes can make 40 bonds in a second. So you appreciate them, don't you? Now that we know how they work, we like to describe them as factories that have 2 floors. In the top floor, the instructions come in and being decoded. And in the lower floor is where the bond is being made. So the amino acids are brought by trucks, which are also molecules, RNA molecules called transfer RNA or tRNA. They are being brought in. And when there is here a triplet that codes for this amino acid, it will come in and be connected to the newly grown protein. And this happens inside the factory. I don't know to draw inside, so please forgive me. But this happens inside in the lower floor. And then the truck will go out empty. The code will progress by 1 codon, 3 nucleotides, and all needs a little bit of energy. And it took us only 20 years to solve how it is, to find out how it is. So once we were able to get the structures, we also understood the function. And we made two little clips. They are both available in YouTube, which are already in YouTube for the last 15 years, and with many, many hundreds and thousands of hits. Nobody complained so far, so I think that what I'm showing you is correct, because if a scientist would have found a mistake, he would shout, yeah? So here you are. The messenger RNA, these are the instructions, is reaching the small ribosomal subunit. Now, I should have told you that a factory that we say top floor and lower floor is actually describing ribosomes. They have 2 subunits, a small one, which is where decoding happens, and a large one, where the bonds are being made. So what you see here is the small one standing, and the messenger RNA, the instructions, is reaching it. And in the cell, the 2 subunits are separated, and they are entities. Each of them is an entity. And when they have to start working, the cell tells them and say initiation factor. So now, initiation, there are in bacteria 3 of them, in human many more. The initiation number 3 is bound to the small subunit. You will see it in a minute. It will be in blue. And he monitors the entrance and the progression of the messenger RNA. You see the blue thing. Now the small subunit is ready to accept the first track, which is a tRNA molecule. And this is also brought by the initiation factor number 2. And when everything is fine, the 2 factors are checking all the positioning and everything else, the large subunit can come, associate, and make bridges, chemical bridges, even in bacteria, 13 of them. So here, the functional ribosome has been made, and we look at it from the solvent side. On top is the large subunit, below is the small one. The tRNAs with the amino acids are coming in, one by one. The ribosome helps, in, out, 40 times a second. They are brought in by elongation factors. We took away now the large subunit, and you can see how decoding happens up. And the newly born protein is tunneling into a tunnel that you can see here that protects it and spans the large subunit. So now you see again the whole ribosome, then protein comes out and folds, and the whole system is waiting until protein is finished, until the gene is exhausted. There is a stop codon, and when there is a stop codon, there are factors like this gold thing, which are recycling and release factors, and everything is over. Isn't it simple? I don't know why we needed 20 years. Okay, so actually what you saw until now is top is the small subunit where decoding happens, and below is the large subunit where the bond is being made. It's called PTC, peptidyl transference center, in here, small and large. I hope you can see. And just to tell you a few things about the tRNA, the carrier, it has two functioning sites. All tRNAs from all bacteria, from all animals, from everything has this L-shaped type, double helical, with a loop that is anticodon, it means the reading, and an end, which is always the same three nucleotides, for those that know names CCA, and the amino acid bound to them. And I just want to tell you that we know the position of each and every atom in the ribosome. Small subunit is now rotating. Ribosomes are made mainly of RNA. This is the gray, and many proteins. Each of them is a different color. I will not go into details. You can see them here. And it's very important that we know the position of each atom, because then we understand what we wanted to understand, how ribosome works and how antibiotics bind it. So you see here the large subunit is rotating, again, mainly RNA, the gray, and many proteins, and if you want some data is here. So as I said earlier, over 40% of the clinically useful antibiotics are stopping protein biosynthesis, and the natural antibiotics are the weapons that one bacteria is using against other. The question that we ask first is how do the tiny antibiotics paralyze the giant ribosome? So ribosome has a molecular weight of 2.5 million Daltons. That's the chemical weight, unit weight. And antibiotics are less than 1,000. They are very clever, the antibiotics. Each of them is binding, is paralyzing one step, like in every factory. It's enough to paralyze one step that there will not be function. So you can see here the small subunit and the large subunit and all these rings. Each of them is representing a family of antibiotics. And look, the decoding, in the bonding, in the tunneling, and in the hinging, this motion. Isn't it clever? Each of them so small and so clever. So once we've got the structures and how the antibiotics are bound, are working, we made also a little clip with the antibiotics, and this is also available in YouTube. You see Edein, this little one was discovered in Poland, and it just sits there and doesn't let messenger bind. That's it, doesn't bind. So small, so clever. Don't have to be large in order to stop. Second one that you'll see in a minute is called tetracycline, which is more powerful or more useful. I don't know what, yeah, okay. So here is Edein, which is, I think, from the point of view of powerful, is very powerful. It's not so useful because it doesn't distinguish between the patients and the pathogens, but it explains a lot. Second one, which is useful, is tetracycline, that occupies the position of the second track, tRNA. just occupies the position. Huh? So small, so clever. The third one that I'll show you is called erythromycin. It paralyzes the large subunit. Until now you saw the small subunit. And it came into use right after penicillin. So it sits at the entrance to the tunnel through which protein will go out. See? That's it. Only very short protein, and that's it. And the last one, from chemical point of view, is the cleverest. And doctors tell me that it's also very useful clinically. And it's called clindamycin. It goes into the bond. Have a look, into the bond. Just sits, finds out where the bond is. So let's talk now about problems. Until now, I only told you stories. Now I'll tell you a problem. Resistance to antibiotics, which is also a natural product, is a most severe problem in modern medicine. So bad, no, it's a way that bacteria from one type is protecting itself against the antibiotics from other bacteria. So bad that the World Health Organization announced that we are reaching post-antibiotic era. And even the World Bank they estimated that almost 4% of the global economy will be lost by 2050 because of resistance. Yet, very few antibiotics are under development by the big companies, pharma companies. They stopped attempt at creating new antibiotics because there is a huge mismatch between the social value, it means our life, and the high expense needed for creating new antibiotics. I will criticize the companies later, but I want to show you data. Yeah, I have critique. This is the way that the resistance grew for 3 different bacteria, and this is the number of new antibiotics. So, before I go on, just my opinion. Do I think that we can combat antibiotics fully? This is unlikely in my opinion, because bacteria want to live. And because they are cleverer than us, at least in terms of survival. So we really have, if we want to talk about new antibiotics, we have to fight against the wisdom of bacteria. Nevertheless, although I wasn't sure we can do it, I wasn't sure that we can beat the bacterial wisdom. We do something in our lab in this area. We hope to contribute something. And the concept is that although pathogenicity is usually not located in ribosomes, it's other mechanisms, we are exploiting unique structural features in the ribosome, in the periphery of it, in order to use it as sites for new antibiotics, because stopping the life of pathogens, it doesn't matter if we stop the pathogenicity or any way else. So first I want to show you, if it works, that we found, it works. We found some regions like this. You see, rotating is the structure, the gray is the structure of normal large ribosomal subunit, and the cyan on it are specific little pieces that are specific to a specific antibiotic, a specific bacteria called Staph aureus. So here you can see it in a standing way, and we talk about these little places that do not exist in what I call harmless antibiotics. So statistically, there are less than 10% of all the other nucleotide RNA, it's all RNA nucleotides, and most of the statistic people told, nah, they won't get anything, it's not enough. And maybe it's not enough, but locally, it's more than enough, because locally, these are specific, unique structural features. Let's focus on this region. Here it is. So all around, the two structures, harmless and pathogens, are the same. But here there is a little piece, additional piece, which is really very small, but large enough for us to assign it as a potential binding site for new antibiotics. So in a model system, we already identified 25 new potential unique sites. And now, by the way, we have already 4 structures of 4 different pathogens, so we can mine all of them. We blocked them one by one, and 16 of them, when they were blocked, they inhibit protein biosynthesis. These sites are not involved in the primary ribosomal activity, hence no pathogens contain genes for their modification. This means that so far we can think about them and exploit them for designed advanced degradable antibiotics, and hence also environmentally friendly. So this is not all, because careful, okay, let's leave this and go to the ecology. And before I do this, just a second. Ecology means that the current antibiotics, all of them have regions that are not digestible. When they are not digestible, they go out with the sewage. And because they are so small, they are not caught by most of the purification systems, so they can penetrate into the irrigation, and then we get them back. Those that we are now suggesting are all made of either degradable, biodegradable, or not able to increase resistance. So before I go, I want to have your attention to this point and this point, which are the two active sites when we look at the large and small subunit. They are all made of RNA. You see, all gray, all gray. So actually the ribosome is an RNA enzyme. We call it ribozyme. And when I ask kids, why do you think it happens? Why RNA has to make proteins? The kids say, the cell doesn't trust the proteins to make other proteins. They will make those that they love. And I think the kids are right. In Hebrew, we call it protectia. And the cell doesn't want it. Also, it's an RNA machine. Even in mitochondria, where part of the ribosome, a large part of the ribosome has been replaced from RNA to proteins. And also, it agrees with the idea that before life, there was an RNA-dominated world. It means this was before life. And when we look at the ribosome, we found that where proteins are being made, it means here, this position, it's all RNA. And it's semi-symmetric. And when we look at the structure, at the sequence, we don't find any symmetry, but the structure we do. Actually, this part that it's structurally semi-symmetric has where the A and the P, it means the two tRNAs are bound. If we look at this structure, it's the same in all ribosomes, from bacteria to elephants. So when we draw one on top of the other, and we have here where we took the coordinates, they are the same. When we look at it from the side, it looks like a machine. When we look at the number, the amount of conservation, it's almost the same in all ribosomes, which means the high conservation means that the symmetrical region is existing beyond environmental considerations. And we think that this is the proto-ribosome. From this, the ribosome developed, which means that in our body there are pieces from the prebiotic time, the proto-ribosome, and still functioning. It makes proteins very fast. You heard it also from Hershkop. And I just want to show you the position of it. Here it is. And here is the way it looks. That's the way the proteins are, the position proteins are being made. You can see it from the side. I don't want to go into detail, just to say that the base of the suggested existence of an RNA-dominated world and on the finding that RNA can replicate and elongate itself, we propose that the proto-ribosome was not only the beginning of ribosomes, but it is the entity around which life has evolved. And in order to prove this, or at least partially prove it, we tried to make it the proto-ribosome. Took us more than 9 years to get it. I want to skip all this. I just want to tell you that a few months ago, we found that the reaction that is made by the ribosome, it means the addition of 2 amino acids, which is usually shown by ribosomologists as these 2 components, can be done, was done in our lab in February. First time. So we actually got a peptide bond. And if any of you want to know details, we did all the types of yeah, I'm finishing. I need only 1 to 2 seconds for the protein. And the question is, what now? So what now? I will not go into all the experiments, just to tell you that we are interested in the emergence of the genetic code, and we think that the initial dipeptides that were made by the proto-ribosome could be the base for larger ones, and those that survived were those that could do specific, like transport of metals, or those that could stabilize the machine, like it is in the ribosome today. So the take-home lesson is that the genetic code evolved together with the ribosome as well with its products, the proteins. And it's the summary that I just showed you. And how does it correlate to Schrödinger? Here is Schrödinger with his book. And the question that he asked, what is first, the chicken or the egg? And we translated what was first, the chicken or the egg, or the genetic code or its products, the proteins. Do you know? Can you suggest? It's difficult, isn't it? We suggest neither. The proto-ribosome was the first, and from this, life was. Before I finish, I want to give credit to...
Can we have a couple of questions?
Just a second. Michael, I want to talk about you. I want to thank the specific people or persons that allowed me to work, although for, I told you 20 years, 15 of them I was the village fool and everybody laughed at me. Nevertheless, Professor Michael Sella, who is sitting here, let me work, although it was called dream. So after he finished his term, Chaim Harari, who is a physicist, he also allowed, and these people encouraged this, but they said, no, it's not clear that Ada will make it. So nevertheless, he let me work. It all started with a very strong collaboration with Berlin, with Max Planck in Berlin, Dr. Wittmann, who wanted really to say how ribosomes are, how the structure is. He died 10 years before the structure came, was replaced by Franceschi and then by Fuccini, but he also was very important in starting a group for us in the synchrotron in Hamburg where we measure. So I had 2 groups, and I want to thank both of them. The group in Hamburg has been finished 14 years ago, but the group in Israel is still working, and this is a collection of people that contributed over the years. Not all of them are now with us. For the young girls here, please, my picture is here, but the one that really runs the group is Dr. Anat Bashan, who is a fantastic scientist and a mother of three, a very good mother of three, so it's possible. Also, Ella is a fantastic mother of three and fantastic scientist. I will not go one by one. I want to highlight Shula, who is here, Shula Weinstein, who is here in the audience, and Tamar. Tamar came 16 years ago for 10 weeks. She's still around. And at the day that I took the picture, she had birthday and she baked the cake. You want to see the cake in detail? Here is her cake. And this shows that in my group, ribosomes are considered sweet. I also want to thank my family, my daughter, who is an MD at Shiva Hospital, and my granddaughter, and my mother, and my sister. All of them were encouraging me, although I was disappearing a lot. And my granddaughter gave me a speech about me. As you know, I'm Noah Ada's granddaughter. As you know, she is a very busy scientist, but she always finds time for me. That's what she said in Paris. And then she gave me a prize. That's not her prize. This is the Nobel medal. Her prize for me is at least as important. The grandma of the year is Ada Yonat. And when I asked her which year, each prize has a year, which year, and she said, if you fail, I'll take it off the wall. It's still on the wall. Girls, it's possible. It's possible to be a scientist and a mother and a grandmother. So thank you, and have a look. In Israel, ribosomes became popular. He is me, and she's his wife. You see, they are dancing, and she has protein comes out of her. She's the ribosome. And this is what happened to me. So Michel Kishka, who is an artist and a writer, he drew me, head full of ribosomes. But if you look, small subunit, like subunit, large subunit, small subunit, full symmetric, and he thinks antibiotics should be here. Thank you.
Are you open to questions? Any questions?
I'm answering even girls.
You said that you found, or I don't want to say thing that you found, but the piece that is the proto-ribosome. Can you think of a way that the ribosome was built around it? Because there's a difference between that tiny little piece and the large ribosome piece.
Of course, it grew around it when the proteins that it could make were longer and longer and started to bind to it. So then there was more RNA to protect the proteins, and then more proteins to bind to the RNA and give it its structure and so on. This is the way I think. Yes, you got it.
Yeah. Okay. Yeah.
About the mechanism of antibiotics, how do they know where to go and how to block those entrances?
About their knowledge, I cannot answer you. But maybe they go to many other places. We don't know. But we do know that when they hit these specific ones, and from the chemical point of view, their specific binding places, binding sites, are fantastically architected. It's like a very, very clever chemist designed them. So when they come to their position, they stay there. Maybe they bind to many other things on the way. We don't know. And those that we are suggesting may also bind to other places, but be not disturbed.
Thank you.
Thank you very much. Ada, thank you once again.